Okay, perfect. Looks like we are live. Hello, everyone, and welcome to the December edition of the COVID Info Commons COVID Research Webinar. We are really excited to highlight lightning talks today from 6 great NSF-funded researchers spanning a number of different disciplines and coming from all around the country. We've got a really great lineup and are so glad to have all of you join us today. We will also be delighted to announce the brand new, first of its kind, COVID Information Commons Undergraduate Student Paper Challenge. There'll be more information about this at the end of today's presentation, so I do encourage you to hang on at the end of the line. One of our amazing students who has been working on the COVID Information Challenge will be presenting this information to you, and we strongly encourage you and your students to get involved. So these webinars are brought to you by the Big Data Innovation Hubs through the NSF Convergence Accelerator Program. And we are, as I mentioned, delighted to highlight the work of 6 different researchers today. 7 different projects, as one of our researchers will be presenting on 2 separate awards that he has received addressing the COVID-19 pandemic. So as I mentioned, we are going to be talking a little bit more about the progress of the COVID Information Commons to date and our upcoming student paper challenge at the end of the hour. So I look forward to introducing you to some additional members of our team to provide further information about all of the great work that this community has been doing to date in addressing the pandemic. But for now, I will just kick things off by introducing our researchers whose names you see here. Just as a little bit of housekeeping, we will be keeping Q&A for the speakers to the end of all of their talks. So please hold your questions or feel free to put them in the chat window. The speakers will be addressing those as they are able to in the chat window, and our student monitor will be collecting several more to read out loud during the verbal Q&A portion later on. So without further ado, I'd like to introduce our first speaker, Foad Hamidi of the University of Maryland, Baltimore County. Foad, we're ready whenever you are.
All right, thank you very much for the introduction and coordinating the webinar. I'm going to share my screen. All right, so my name is Foad Hamidi. I'm an assistant professor in information systems at the University of Maryland, Baltimore County. And it's my pleasure to present this research on behalf of my colleagues, also at Digital Harbor Foundation, Andrew Coy, and from Project Waves, Adam Mohammed. This is a collaborative rapid project, and it's funded obviously by the NSF. So the topic of the project is responding to COVID-19 with community internet. So across the United States, there's a significant number of people who lack broadband access to internet in their households. Depending on where you look, the number varies. So according to the FCC, a very conservative estimate is about 21.3 million people. And then Microsoft also has estimated about 151 million people. So there is, regardless of the exact number, there is definitely a significant number. And in Baltimore City, where our project is located, according to the 2019 US Census, there was 28% of households that didn't have access to broadband internet. And also the Baltimore City Schools did a recent survey in response to COVID, and about 24,000 students don't have access to devices or the internet connectivity during this time. So this is obviously a problem even in the absence of COVID, but of course with COVID there is a lot of opportunities, including employment opportunities, educational opportunities, and also access to opportunities for social interaction that people are missing out if they don't have this internet accessibility. So in our project, we are investigating an effective and efficient community-based approach for setting up free high-speed internet access in urban settings. So we're looking at both the technical and the social aspects of this question. On the technical side, we are looking at how to set up mesh networks in an urban setting. So a mesh network essentially connects to the backbone internet using a wired link, and then you can distribute routers, wireless routers across a geographical space and quickly set up internet activity or network connectivity. In our case, it's the internet across the community. So our project involves three phases. So first, it involves setting up points of presence. So in the city, we partner with organizations that have basically points of access with good visibility. So there should be basically a clear line of sight between an antenna and the routers that we want to connect with. And these include very tall buildings in our city, and also, for example, buildings like churches and so on that can provide this access for us. And the city has been very generous. Our community partners have been very generous to kind of set up that infrastructure. The next step is going into the community and setting up routers at each house and kind of setting up so that to make sure that the system is compatible with the household. And then finally, creating online trusted information resources so that our community members can access these resources in a timely manner once they have the connectivity. So so far, we have received some very positive feedback from the participants. From a research perspective, we're conducting both surveys and also interviews to understand what is the impact of having this access. And I just have a couple of quotes here. Quickly read them. Internet connectivity is a necessity. Everybody needs it. Everybody should have it. And another participant said, COVID is a stagnating life as we know it, and having this internet is allowing life to go on. So we're looking forward to continuing the work, and as we're going along documenting what are some strategies for effective deployment, and also what are some barriers and how we can overcome them in the face of pandemics in the future. Yeah, I think that wraps up my talk. I hope that I was in time. I think I have one last slide. So there's a lot of people involved here. I want to thank my students and also amazing community partners, Project Waves, working on the ground, setting up the infrastructure, and also the Digital Harbor Foundation, iteratively creating online resources for the community and getting feedback from them to see what is needed. And of course, I'd like to thank the National Science Foundation for their generous award for us to be able to do this work. All right, thank you very much for organizing this talk, and I'm happy to answer questions afterwards.
Thank you so much, Foad. Really appreciate you sharing this. Really appreciate the work of you and your whole team disseminating these resources. That's amazing. For those who have just been joining, hello. We're so excited to have you here. Please add any questions that you may have for our speakers in the chat. They will be keeping an eye on that, and we will save all questions for the end that are not addressed there. So our next speaker is Aron Laszka of the University of Houston, speaking on addressing transit accessibility and public health challenges due to COVID-19. Aron, we're ready whenever you are.
Thank you for the introduction and the opportunity, and welcome everyone. Please let me know if you cannot see the slide.
I can see it, it's great.
Perfect, thank you. My name is Aron Laszka, and in this talk, I will talk about the significant challenges that public transportation faces due to COVID-19 and how our research project addresses these challenges. And this is a collaborative research in collaboration with Professor Abhishek Dubey from Vanderbilt University, as well as the public transit agencies of Nashville and Chattanooga, Tennessee. I would like to begin by emphasizing how important public transportation is to many communities. Public transit services connect people with access to employment, education, and other public services, which they would not be able to access otherwise. Unfortunately, COVID-19 has significantly disrupted the operation of many public transit agencies and have created a lot of challenges for them. These challenges include, on the one hand, resource challenges. For example, due to social distancing requirements, vehicles have to operate with reduced capacity. So a vehicle that could, under normal conditions, transport 30 or 40 people now can only carry about 10 or 15 passengers. And of course, this is a necessity due to social distancing requirements. To make matters even worse, a lot of transit agencies are also struggling with driver unavailability. A lot of drivers are unavailable due to isolation or quarantine requirements, or maybe they're in an at-risk population. So at the end of the day, resource challenges are really significant. Transit agencies have to operate fewer vehicles at a lower capacity, which has significantly reduced transit accessibility. And unfortunately, these resource challenges are made even worse by data challenges. Due to COVID-19, how people use transit has significantly changed. Ridership patterns have changed both temporally and spatially. And in order to respond to these changes, transit agencies need new data, but it's really difficult to collect this data at a short notice. And to make things even worse, a lot of transit agencies have switched to fare-free operations, which not only decrease their income, but also basically not make them unable to collect data from fare collection. Our research project addresses these challenges through three main thrusts. First, we perform data analytics. This is based on data collected from Nashville and Chattanooga through a variety of means, including automated passenger counters as well as manual counting. Using this data, we perform data analytics. The purpose of this data analytics is to help transit agencies to estimate ridership patterns, see which lines need more resources, where transit agencies could save resources, as well as predicting ridership using machine learning models so they can optimize proactively. We're also planning to develop applications, mobile applications for passengers that will provide guidance to passengers on how to travel more safely and more conveniently by avoiding crowded vehicles. In our second thrust, we provide guidance to transit agencies. We're helping them with operational optimization. This includes both proactive optimization, using predictions in order to optimize vehicle dispatch, routes, and schedules proactively. The goal here is to maximize transit accessibility while at the same time maintaining social distancing requirements and considering other accident challenges like disinfecting vehicles regularly. And we're also providing on-demand optimization for dispatching paratransit vehicles, which are really important since a lot of the passengers on these vehicles are from at-risk populations. Finally, in our third thrust, we're applying computer vision to collect ridership data using onboard cameras and video analytics, as well as monitoring passengers from compliance with requirements such as social distancing and wearing facial covering. Finally, I would like to present some preliminary results around data analysis, which I hope you will find interesting. First, we performed temporal analysis. We looked at how ridership has changed over time. Now, one obvious finding is, of course, that ridership has significantly decreased. In the plot in the upper right corner, you can see how ridership, the red line, has decreased significantly in this spring and remained consistently low. More interestingly, ridership has changed temporarily within a week and within a day. Specifically, we see a much larger decrease on weekdays, as well as during early morning and afternoon hours, typically when people commute, which suggests that most people don't use transit for traveling to work or from work, while they still use transit for other purposes. Finally, we also perform spatial and socioeconomic analysis. For example, we have investigated in which areas there was a most significant decrease in ridership. Interestingly, we have found that Areas where there are a lot of retail and shopping opportunities have seen a much larger decrease, which is of course not surprising. We have also considered socioeconomic factors, for example, income levels. As you can see in the bottom right corner, we have found that areas where residents are generally lower income have seen a lower decrease in ridership. We have also found similar correlation between other socioeconomic factors, for example, housing values, as well as certain races. And with that, I would like to thank you all for your attention. And I would like to ask you to please enter your questions in the chat or contact me or my co-authors by email at a later time. Thanks, everyone.
Thank you so much, Aron. I encourage our presenters, please feel free to drop your contact info in the chat if you'd like people to follow up, just so they're able to grab that a little more easily. Moving right along, our next speaker will be Guan of Claremont Graduate University. Please go ahead whenever you're ready.
Thank you, Katie, and thank you for the opportunity. So I'm presenting on behalf of a team between Claremont Graduate University and University of Kansas. My colleagues, Chining Chang, he is actually on the Zoom too. So he will help with answering questions if you have some burning question that you are asking. So our project focused on the electronic mentoring among engineering graduate students during the COVID-19 pandemic. And it was funded by the NSF Rapid Program, as many other presenters here. So today I'm gonna just focus on a subset of the study that we conducted back in June. So we are focusing on 566 engineering graduate student sample in our study. It's part of the national study of STEM faculty and students. We do have like more than 1,000 faculty samples and 3,000 undergraduate students. So we are still digging into the data. It is cross-sectional, and we are thinking about to do a follow-up survey and interview in the spring to see what's going on. So the sample was diverse. One thing is it was a difficult time. We will try to get more minority students, but at least we get about 14% of Black, Hispanic, and Native American students. And in terms of gender or disability or SES, is kind of like quite diverse samples that we have here. Okay. So first, we're going to present three sets of student outcome that we care about. First, we in the spring, at least a quarter of student, graduate students say that they might be delay their graduations due to the pandemic. And we saw that PhD students tend to report higher level of that, higher rates and low SES compared to master student. And in terms of job search, self-efficacy or confident in finding a job that they are prepared for or they are qualified for, we see overall across the board, there's a decline in the confidence. And what is concerning here is female student, low socioeconomic status student, student with disability, they all have, they reported a significant lower level of all these indicators. And then another set of indicators, mental health, we use the same set of depression and anxiety indicator used by US Census. There's a household pulse survey conducted in from April to July. We are able to compare our sample to the general population. So good news is our graduate student sample, they report better than the general population because they are not the frontline workers or they less likely come from low income family or what. But within this group, again, underrepresented group, student, female, low SES and student with disability and international students tend to report higher level of those mental health issues. So this is something that we would like to highlight here. So our focus is, can e-mentoring address some of these challenges? So one pattern that we saw, and I think this is common sense, we know during the pandemic, most communication are being on video conferencing or email. So it's more likely that, and much fewer hours in face-to-face, and it makes sense. So this speaks to the experience that we have in academia too. So we collect a lot of survey skills indicator that we perform structured equation modeling here to look at the relationship across different indicator. So what we found here is e-mentoring frequency predict both instrumental support and psychosocial support from the mentor. So instrumental support tends to be like their mentor help them to complete their assignment, or their writing or presentations, more instrumental, more technical. And psychosocial support is more about address their concern, anxiety, talk about their future career options, something like that. So we see that the more frequent they interact, even through electronic platform, would have positive impact. And those lead to higher level of mentoring satisfaction, and also lead to more positive outcome that I just mentioned in the past few slides. And we also look at how e-mentoring attitude would predict e-mentoring frequency too. So we asked them whether they think e-mentoring through email or video conferencing would be more effective or equally effective as face-to-face. So those who have more positive experience or perception of e-mentoring, they tend to interact more too. And I think this is, is predicted by the theory. So, and lastly, Nick Chining Chang, my colleagues here, his area is in individual development plan. So we were interested in those graduate students who have IDP before pandemic. Do they interact more with their mentor? Indeed, we found that that is the case. Among our sample, there are about 30% of the students, they say that they have IDP with their current mentor. And so this finding is very important. That means that infrastructure, a program like this before a crisis like pandemic or any other crisis would facilitate or promote positive interaction and outcome when we have any crisis coming up. So lastly, we want to talk about this is that mentoring while social distancing is challenging, because in our sample, in our study, both from faculty and student, generally they think face-to-face is still more effective than e-communication. Back then, this is more June. And we hope to see this attitude will change. Now they are being forced to do a lot of e-communication. Probably they are getting used to it. But even with the sample that we have from June, about 25 to 50% of faculty and students, they believe that e-communication could be equally effective as face-to-face in certain areas, including especially helping them in terms of writing skills or preparing their job application, something like that. So this is something that promising, that important. So with that, I want to thank you, and thanks for the opportunity. Yeah.
Thank you so much, Guan. Really great to see these important issues being addressed. I know it's a stressful time for a lot of folks. So in terms of our next speaker, I'm excited to introduce Gloria Oporto of West Virginia University, who will be talking about building a prototype of an improved medical mask. So Gloria, whenever you're ready. There we go. Gloria, if you are speaking, you are on mute.
Now, yes. I'm sorry. Yes, I was on mute. Sorry, I was asking if everybody can see my presentation, and I believe so.
Yes.
All right. Good afternoon, everybody, and thank you very much for giving us the opportunity to present our research titled Prototype of a Medical Mask Using a Novel Antimicrobial Antiviral Biofilter Material. I am Gloria Oporto, Associate Professor at the Wood Science and Technology Department at West Virginia University. And here is the team that is working in this project. Dr. Rakesh Gupta, copy I, Dr. Edward Sabolsky, copy I as well, Dr. Sushan Agarwal, Dr. Jonathan Boyd, Dr. Rosie Santos, and our grad student, Kevin Sivaneri. So let's see, let me move this. There you go. The synergetic efforts to perform this project are coming from 3 different departments and colleges at West Virginia University. The College of Agriculture, Natural Resources and Design, the College of Engineering, and the College of Health Sciences. So now, considering that there is an important increase of healthcare-associated infections in hospitals due to the increased load of deceased patients and the lack of proper and effective protection for the medical community. The common personal protection equipment are disposable, non-degradable, single-use items, vulnerable to penetration by microorganisms, and also the common type of particulate filtering face piece respirators are made of non-degradable, non-woven polypropylene fiber and removes 95% of airborne particles. So our main goal is to develop a novel biofilter that will be a non-woven material prepared with polylactic acid fibers in combination with nanocellulosic fibers, which in turn will be coated with copper nanoparticles. So the final material will be able to remove 99.999% of airborne particles and will have antimicrobial properties, and those are going to be provided by the application of copper nanoparticles. So the central hypothesis of this project are that by coating efficiently and quickly bio-nanocomposite filaments, polylactic acid plus cellulose nanofibers, with copper nanoparticles, we will reach the effectiveness of these particles towards killing 99.999% of bacteria and viruses. And the second is considering the diameter of the nanocellulosic materials are smaller than the common size of the new coronavirus COVID-19, which is spherical with a diameter of approximately 125 nanometers. So it is expected that the viruses can be retained in the biofilter prepared with an optimum and well-distributed amount of nanocellulosic material. So to move fast, we printed a prototype of a mask in a 3D printer using two different filaments, polylactic acid that you can see here in white, and polylactic acid reinforced with wood particles. So in this design, we are focusing our attention in developing our biofilter that will be incorporated here. So in the pictures on the right, you can see some examples of 3D printed specimens prepared with polylactic acid and wood. However, our attention right in this presentation will be in the biofilter that will be here. So in the preparation of our biofilter, we are using two approaches, electrospinning and force spinning processes. In the electrospinning process, electric forces are used to produce the fibers. And here you can see some examples of polylactic acid fibers produced, right? And here you can see different magnifications of these fibers. And it is expected, right, that the porosity that you can observe here can help us to for a better incorporation of the antimicrobial copper nanoparticles. So in the case of force spinning process, centrifugal forces are used to generate the fibers. And here also you can see the final porosity, right, generated for this specific process. So we have been also following different approaches in order to incorporate copper nanoparticles on the fibers. And one of them is using a master batch copper polylactic acid provided by a Chilean company. And here you can see also that the particles right on the surface of the fibers correspond to copper that we analyze and of course confirm that there are copper nanoparticles on the surface of these fibers. So regarding to testing, we are using again a 3D printing process to optimize a filtering device that will be used for all the specific tests that are related to our biofilter in terms of breathability, filtration, and fit test. So finally, our ongoing work is combining our polylactic acid fibers with nanocellulosic fiber and copper nanoparticles and determining their antimicrobial properties. And almost in parallel, right, we are starting with the full characterization of the biofilter following standards presented in this slide. So with that, I am finishing. Thank you very much for your attention, and thank you, NSF, for providing funds to execute this project. So thank you again, and if you have any question, I will be on the chat answering, and my email address is also presented here. Thank you again.
Thank you so much, Gloria. I really appreciate the talk. So our next speaker will be Janet Williams of University of Idaho. Janet, we're ready whenever you are.
Thank you. And can you see my screen?
Yep.
Yes, great. So thank you for the introduction, and thank you to the organizers for allowing me the opportunity to present about our study today, which was funded by the NSF Rapid Collaborative Research Program. The title of my talk is COVID-19, Human Milk in Infant Feeding. I'm presenting today on behalf of a wonderful group of collaborators who I have the honor and privilege to work with. I have collaborators here at the University of Idaho, and I believe Dr. Ryan Pace is on the call as well, at Washington State University, University of Washington, and Tulane University. The goals for our study was to determine whether or not if human milk is a potential mode of transmission for the SARS-CoV-2 virus, and also to examine antibody kinetics in breastfeeding and non-breastfeeding women and their infants. So to take a step backwards, why is this study important? Well, we knew back at the start of the pandemic that some viruses like HIV can be vertically transmitted or transmitted via human milk. And so at the start of it, there was a lot of uncertainty. And so the initial response to the COVID-19 pandemic was that at times and in several locations, women who were diagnosed with COVID-19 were separated from their infants and also discouraged from breastfeeding. But this has important ramifications, as you might imagine, because human milk not only contains important nutrients and bioactive factors that are important for the growth and development of infants, but also it contains important immune factors that help protect from disease. And so research needed to be done to determine what were the potential risks and benefits of breastfeeding. So we designed a study in which we recruited 25 breastfeeding dyads or mother-infant pairs. And also we were hoping to recruit 25 formula feeding dyads as well. Mothers, we recruited mothers who were diagnosed within 7 days of having their COVID diagnosis. We have met our target for the 25 breastfeeding dyads, but unfortunately we have not met our target for the formula feeding dyads. And so we have pivoted, as we often have to do in these COVID times. And so we are now recruiting 25 breastfeeding dyads with the mothers who are healthy and who have not been sick. From these moms and from their infants, we are collecting milk, breast swabs, blood spots, stool samples that are collected 7 times over 2 months, as well as information about maternal and infant health, infant feeding practices, and related behaviors. Our ongoing and planned analyses for these samples are that we will be examining the milk, the breast swabs, and the stool for presence of the SARS-CoV-2 RNA, as well as looking at antibodies in both the milk and the blood, and then relating this with the information that we're also collecting to infant feeding mode as well as maternal COVID status. These analyses are ongoing as well as our recruitment is ongoing, but to present just a little bit of the preliminary data or some of our preliminary findings, I can share with you that from the milk samples, we have not detected any SARS-CoV-2 RNA. And this is consistent with previous studies that have now since come out to show that milk does not appear to be a vehicle for the transmission of the virus. This is good news for moms and infants. And so the CDC and the World Health Organization has changed their recommendations such that it is now recommended that moms and infants that moms should continue to breastfeed even if mom is COVID positive, but using appropriate preventatives like washing hands and wearing masks. So I appreciate your time. And once again, on behalf of all my collaborators, I thank you for your attention. And I thank you to NSF for providing the funding for this information. And if you have any questions, I have my email here, as well as emails for the 4 lead PIs at each one of the institutions. Thank you.
Thank you so much, Janet. Really important and interesting questions being addressed here. So I will, with that, hand things off to our final speaker, John Yin of the University of Wisconsin-Madison, who will be discussing two separate awards that he has received through NSF addressing COVID-19. So John, whenever you're ready.
Great, thanks a lot, Katie. I think you're able to see my slide, but I maybe need to go to project mode. Can everyone see that and hear me?
Yes, thank you.
Great. So I'm John Yin. I'm a professor of chemical and biological engineering at the University of Wisconsin-Madison. I'm also with the Wisconsin Institute for Discovery, and I have the honor of sharing some of our work. This is actually ongoing work for the last 15 years that we've pivoted to bring over to coronavirus. It's about turning the dial on coronavirus infections. We have two awards. One of these involves turning up the dial for infections, and one involves turning down the dial. I'll try to explain what that means. I didn't do any of the work. The people who did the work are all here, past and present, many of them now in industry or academia. I think two of my co-workers, Nan Jiang and Huicheng Shi, are on the call with us today. So these are the people who do the actual work. To help you understand where I'm coming from, I have to tell you how we quantify infectious virus. So there's a standard way to quantify infectious virus. So we have a virus stock solution here that we'd like to figure out how much virus is actually in there. Usually there are millions or tens of millions of particles. We would like to figure out how many particles and count them. And the way we do that is we do a series of dilutions, known dilutions. It's kind of watering down the drink until we get a few tubes that just have countable numbers of particles. Those tubes, known volumes from those high dilution tubes, are put on monolayers of cells, shown here in the red, and overlayered with agar gel. And they are allowed to reproduce. The virus is spread locally and kill the cells. Then we stain the cells blue, and wherever this virus has killed the cells, there's a little hole there. The hole is called a plaque, a region of dead cells caused by a single virus, which we can now see with the naked eye. By counting those holes or plaques, we can deduce, knowing what volumes we had and how much we watered down the original stock, we can figure out how many particles we had in the original stock. So this is a key tool that we use to quantify infection and that we will use to characterize how we turn down or turn up viruses. So the first thing we did here was to try to explore ways that we could get more signal out of this kind of assay. And what we did was, instead of overlaying with agar, so here I show on the no flow cases, these are sort of standard plates that you might see, Petri dishes where they have plaques and you would count those plaques. We carried out the infection in the presence of liquid rather than agar overlay. And we found that if you don't touch the plates, spontaneous flows arise, outward radial flows, and create comet-like morphologies of these plaques. So there's flow that's automatically happening in there that's helping to spread the virus. And for plates that have the same level of virus, we see a lot brighter signal. So we have a much higher signal to noise. We've done some theoretical models, computational models of this process. In essence, we said this might be a useful tool to characterize drugs. So what happens if you add drugs against the virus to this kind of signal? Here in the top left here, we have a sample that has no drug in it. And then we have 5 other samples that have different degrees, increasing amounts of drug. And what you find is the fireworks dim as we go to higher level of drugs. That means we have less and less virus spreading and less infection. We can quantify that here. And on the plot, you see a comet enhanced, flow enhanced comet assay versus the plaque reduction assay, which is currently the gold standard. So our assay is about 20-fold more sensitive and faster to run than the existing plaque assay for drugs. So this is something that we patented 5 years ago, and with the NIH funding, NSF funding, we are now going on to adapt this assay to test drugs against coronavirus. That was turning up the infection and facilitating the spread. We're also interested in turning down the infection. How do you dial down virus infections? It may be happening in nature. Here are 3 scenarios. The first one is the virus infects the cell and it makes a bunch of virus particles. Typically for coronavirus, it makes about 100 coronavirus particles per cell. Among those virus particles, there may be some defective ones. So the defective ones, this orange, as I've shown in the orange here, if it gets into a cell, it does not make anything. It's not able to grow. However, the defective particle retains some aspects of the virus, the ability to use the virus replication machinery. So if both an intact virus, shown here in blue, and the defective virus, shown in orange, get into the same cell as a co-infection, then you can have the defective virus reproducing, stealing resources from the growth of the normal virus. So these defective particles are called defective interfering particles because they interfere with the normal growth of the virus. They've long been known since the 1950s, and in the 1970s, I've taken this abstract from this review article, very prominent review article in 1970. So 50 years ago, people were speculating that these might play some role in viral diseases. Building on that, we thought it would be interesting to try to quantify interference. How do you quantify interference? Well, we set up something like the plaque assay, and the details here are not so important, other than we had to make some dilutions. And instead of providing live cell to viruses the way we do for the plaque assay, we have to provide infected cells to defective particles. That's what the defective particles prey on. So if I go to the next slide, we can see the data such that as we go to higher levels of these defective interfering particles, going from left to right on the x-axis here, the virus production drops and then comes back up. If we look at the production of the dips of the defective particles, how they depend on their own input levels, we have this other kind of behavior that's increasing up to a point and then dropping off very, very sharply. So the key point here is that dips exhibit a complex behavior with respect to their ability to inhibit virus growth, and also with respect to their ability to influence their own replication. So to better understand this, we have studied a different kind of virus, a rabies-like virus, and we've engineered 2 kinds of virus. One that is carrying a red fluorescent protein. So when it infects cells, it turns the cells red. And one that's a defective particle that carries a green fluorescent protein. So the scenario is here. There are 2 different viruses. One is the virus, red, and one is the defective virus, green. And what we do is infect a single cell with both of those and then watch how they spread over several generations. So in this dish, you can see at 3 hours post-infection, you see no fluorescent protein. And if you watch the top right-hand corner, you'll see the time ticking off as we take images at various times. This is 300 microns, or about 1/3 of a millimeter. Okay, so watch the timer and watch the patterns. This is our pandemic in a petri dish. So millions of cells are being infected from that single one. Virus particles and defective particles released from that cell are spreading over this time period of less than a day or so. So we have used this to study in depth what's happening at the single cell level. And these sorts of patterns then you'll see are quite complex. It's really a predator-prey behavior in the sense that the defective virus is preying on the infected cells. If we look at the extent of normal virus spread in the absence of any defective particle, we can see the red expanding. In the presence of defective particle alone, there's no infection. But in the presence of both, we get a co-infection spread that is inhibited relative to the normal virus. So we wonder, could defective coronavirus be used to treat COVID-19 in a way to inhibit or slow down the spread? So today I've told you about two cases where we dial up infection spread, and this is good for antiviral testing because it gives us a more sensitive assay. And I've also talked about dialing down infection. There are natural particles that arise from these infections that can inhibit the growth and spread of viruses. We hope to investigate this for reducing the severity of COVID-19, I'm sorry, that should be 19. If you'd like to learn more, I'd be happy to respond to chat questions or email. Thanks very much.
Thank you so much, John. Really appreciate this overview of all the work that you're doing on COVID-19, you and your team. So thank you. Now that our speakers have given our lightning talks, I've been seeing folks having a good back and forth in the chat. Benji, would you be able to read out any questions that have not yet been addressed in the chat for researchers to answer?
Yes. So this is a question for Janet. The question says, since secretory IgA is the key Ig found in milk and can neutralize the virus, your finding is reassuring. However, IgA deficiency in the #1 PID worldwide with 0.1 to 0.7% individuals is affected, depending on ethnicity. Have you included or plan to include any IgA deficient mothers in your study to see if the virus is present in milk, and if so, maybe transmitted to the infant?
Hi there. Thank you. That's a great question and a great thought. Unfortunately, at this time, we're not actually specifically targeting, different groups. We are just recruiting, all women who, any of the moms who have contacted us in regards to recruitment. So as we go along, we are measuring the antibodies. And so we will be able to maybe see those who have low antibodies in their milk and be able to relate that, but we're not specifically recruiting for that criteria.
Thank you so much, Janet. Appreciate it. I've seen a couple of questions for Gloria Oporto, but I think that she has caught up with those at this point. Benji, are there any additional questions at this time?
No.
Okay. Thank you guys for sharing your questions. I would encourage folks to add any additional questions in the chat if you are still thinking of things, and our speakers will be able to continue to address those. I see that Dr. Oporto has added her email address as well in the chat. I do encourage folks to get in touch with the speakers. I know we've had many successful collaborations, including among some of the people who are joining us on the call today, previous speakers from previous events who have gotten connected with great collaborators that way. So glad to see this back and forth happening. So on that note, I will share my screen once more, and we will talk a little bit more about what's going on at the COVID Info Commons with an exciting new announcement for you all. So I will hand things off to our Executive Director, Florence Hudson, to provide a little more context about the Commons and our upcoming Student Paper Challenge. Florence?
Thank you so much, Katie. And it says you're sharing your screen. I don't see the chart. I wonder if it's me. But I'll just start talking while you look at that. Or I can do it if you prefer.
Let me give that another shot before I give up.
I know, right?
Okay, do folks see the screen at this time?
Not yet. Why don't I project? I'll just share my screen. If you want to stop sharing.
Sure.
Thank you. Okay. We always have backup plans, we've learned. You can't even imagine how many backup plans we have. Belts and suspenders, as we used to say. So thank you so much, everyone, for joining us, and especially for the incredible researchers. The COVID Information Commons was announced in May, thanks to an NSF award from the Convergence Accelerator Program. And we kicked it off in July with our MVP, and it grew immediately into a COVID Info Commons community. We've had some updates since then. It provided 2 search tools at the beginning. We announced a 3rd search tool on October 30th, which is this COVID Awards and PI database. And we have expanded from 700 awards to about 990 awards. And then the PIs are able to send in information, and we enrich the information around their award with their collaboration opportunities, their ORCID ID, and other opportunities that they share. We're delighted with the growth of this. Please join us for more of these webinars. And we've seen incredible collaboration through the KIC. There's one researcher in New York who was contacted by a researcher in South Korea who brought in their mentor from the UK. And now they have this global collaboration going on. Another one of our presenters in September said that since they presented in September on the COVID Info Commons webinar, they've had 30 people reach out to them to collaborate. So this is a great opportunity to collaborate about so many topics like you saw today. Think of what you saw today from, breastfeeding, in infants and moms, which is just so very real. And you've been able to change practices and processes already, which is amazing, to transportation, to, dialing up the virus and dialing down the virus. And it's wonderful how you can each find someone to collaborate with in this community in your domain. So thank you, everyone. And I really want to thank Katie Nam for putting all these webinars together. She's done a wonderful job collaborating and helping make this come to life for all of us. Now, even more exciting is that we have one of our students, and at the Northeast Big Data Hub and the other 3 hubs around the country, we build community and around data science. And one of the things we have is a few students on our team at the Northeast Big Data Hub. Benji has been helping with the, which you probably have seen, with the questions. He's been live streaming. Helen Yang is going to present next, and she's been kind of our COVID Information Commons like sensei, and she's been helping us cleanse it and develop it. And she and Haley is on the line as well. And today Helen is going to announce our brand new COVID Info Commons Student Paper Challenge. She developed this whole thing, and she's going to announce it today, and we're so proud of her. And we're really hoping that you all can participate as a mentor or a judge, or share it with students who can participate and create papers. So Helen, I'd like to pass it over to you.
Thank you so much for the introduction, Florence. Hi, everyone. My name is Helen Yang. Like Florence said, I'm an assistant at the Northeast Big Data Innovation Hub, and I'm also a 3rd-year undergraduate student at Columbia University studying mathematics and statistics. So I've been supporting the development of the COVID Information Commons over the past several months, and I'm really excited to help announce today our newest community initiative, the COVID Information Commons Student Paper Challenge. So the goal of this challenge is to inspire students to learn about the diverse and collaborative ways researchers have been addressing the impacts of COVID-19 to help students form their own insights on the trajectory of and next steps for COVID research, and ultimately have the opportunity to apply what they're learning in their classes to make an impact towards a global issue. So to summarize this challenge, we're inviting all undergraduate students across the world to write a 4 to 5 page paper on a trend or question related to COVID-19 research. And we're asking them to leverage the KIC resources to develop their ideas. So and winning papers will be published on the KIC website and shared widely throughout the community. And authors of winning papers will have the opportunity to present their insights at a future KIC event. So yes, at the top of this slide, we have a link to the web page that we've built for this challenge. You can see the link right there, and you can also navigate to it from the menu of the KIC website through the drop down that Florence is pointing to right now. Thank you, opportunities and resources, and then KIC Student Paper Challenge. So I'll briefly walk through this webpage and go over some of the important details about this challenge. And we of course invite you to look through this webpage as well and share it with your communities. So if we can move to the next slide, please. Thank you. So at the top of this page, we just have an introduction to the challenge and also the COVID Information Commons, along with an email for participants to contact with any questions about the challenge. And this email is info@covidinfocommons.net. And if you have any questions remaining after the webinar today, please feel free to reach out to this email as well. So right below the introduction, there are links to 2 participation forms, one for undergraduate students and one for mentors and judges. So to start with the students, by filling out the participation form, students will be notified about upcoming deadlines and new resources that they can use while developing their papers. The student participation form will be due on February 1st, along with the title and abstract for the paper. So we're also inviting all researchers and experts working on COVID-19, like many of you, to sign up to participate in this challenge and share your expertise as mentors or judges. So we'll be scheduling sessions in January, February, and March for mentors to meet with students to share their research experience and to help students develop their papers. Judges will work with the project team in March to develop specific evaluation criteria for the papers and help review submissions in April. So the deadlines to submit the mentor or the judge participation form are listed under the key date section, which maybe we can move to the next slide to see. Yes. And to clarify, it's the same participation form for mentors and judges, but different deadlines because we're hoping to start these mentor sessions in January. So please consider signing up as a mentor or a judge for this challenge if you'd like to work with students interested in learning about your work and really continue to inspire the next generation of researchers. Okay, so on the next slide, we've broken down the details of this challenge into a couple of categories, and I'll go over them at kind of a high level to cover some of the important information. So on the next slide, we can see a brief description of the challenge, again, introducing students to the NSF COVID Awards, and also what we're kind of looking for them to do in this challenge. So again, a 4 to 5 page double-spaced paper discussing a specific trend or topic that could contribute to future discovery and innovation around the COVID-19 pandemic. So in terms of eligibility, since this is our first challenge, we are asking, we're inviting all undergraduate students to participate. And in the future, if this challenge is successful, we'd be happy to expand the effort to include students of all ages. So great, and if we can move to the next slide, please. So we've listed a couple of example approaches, but truly we want to keep the prompt as open as possible and really encourage students to find their own interests related to COVID-19 research. So for example, students could discuss differences in regional research related to COVID-19. So for example, what have researchers on the East Coast been really focusing on compared to the West Coast? Or students may, as an example, look at a potential cross-disciplinary area of research. So we've listed a couple of resources, both KIC resources like the Research Explorer tool, which has machine learning generated maps of the data around COVID-19 research. So you can see how kind of where research has been focused, and there are a lot of really cool search mechanisms that you can play with there. We also have, of course, the KIC NSF COVID Awards and PI database. This is our newest database launch, and there many of you who are working on COVID research projects funded by the NSF will be able to find your projects there. And this database also includes information about the PIs and any external information that the PIs want to share about their projects. And as a reminder, please fill out a PI survey if you would like to add any information to your profile in this database. And of course, students are welcome to use any external additional resources as long as they're all cited and accurate. Great. So moving forward, we have a frequently asked questions section, and these questions range from eligibility to formatting of the paper. Feel free to read through these, and if you have any additional questions that you can think of, please again direct them to info@covidinfocommons.net. So, and we might be able to add your questions to this frequently asked questions section. And finally, we invite all students to join the KIC community, and they can do this in a couple of ways. So first, we invite all students to sign up for the KIC community newsletter to be notified of new research, news, and opportunities. They're also free to join the KIC Slack community and kind of see all the conversations that have been going around COVID collaborations. So finally, we invite students to register for the next KIC Community Lightning Talks and Q&A webinar. Those are happening in January and February, and really just be inspired by a lot of the research that's happening and to listen to some of these amazing presenters like those who spoke today. So if you have any other questions, I think we can take a couple now, or again, feel free to email us with any additional inquiries. So I see a couple of questions in the chat. Great. So can high school students who are researching with a mentor apply? That's a great question. And I think for this challenge, we are limiting the participants to undergraduate students. I don't know, Florence, would you like to provide another perspective on that?
Sure, because we're very community focused, depending on how many requests we get for things like this, maybe we could add something for high school students, but we really wanted to start with undergrads and see what goes from there. But I would say, why don't you send us an email to info@covidinfocommons.net, and then we'll keep track of that and then let you know if we can at this point plan to expand it. I prefer to do things once in a row, as I used to tell my team, when I was at IBM for many years, and then we can grow from there because we really want to figure it out. And also, if it's going to be high school students, I was thinking a little bit about what type of mentors would we need? Would that be different? Would they be high school teachers as well as maybe higher ed professionals or other professionals? So we actually have a colleague on the phone who's done some high school programs regarding regarding COVID before Dominique out at USC. So we could potentially add that, but we decided to start with just undergrad. So we'll gather, the cards and letters and emails that come in, and then we'll see if we get enough people that are interested and want to help with that. We could see maybe adding that after we're done with the undergrad one.
So thank you for that question. And again, please email us to tell us a bit more about what you're thinking, and we can provide a bit more feedback. So I see another point in the chat that a data-driven challenge might be an interesting idea. And definitely, I think we wanted to start this challenge as a paper challenge to really be able to encourage as many students from different academic backgrounds with different interests to really contribute their ideas. But it would be great, students could definitely write a really data-driven paper and kind of conduct their own data analysis and explain that through the paper.
Great answer. Yeah, their paper could be about data. That would be great, especially since we're the big data hubs. We love that idea. And then, as we said, send in your cards and letters to info@covidinfocommons.net. And as we proceed, we can look at what the follow-on challenges would be if this one is successful. So if you can help us make this one successful around papers for undergrads, that would be great, because then that will give us impetus to say, oh yeah, this worked out great. Let's look at how to expand it based on community input. So we really appreciate your excitement about this, because we're really excited about it too.
Thank you so much, Helen, for all of your work in, developing this and working with us and the whole COVID info community. I think that, we're really excited to get this off the ground, and we do hope that those of you who are on the call today help spread the word, and, whether you get involved or encourage students to get involved, we would love to see folks participating from your neck of the woods. So I will just wrap up here. Our next event will be held Wednesday, January 13th in the new year at noon. We'll have another great roster of speakers, and all of that info is available at the bit.ly that I shared there and shared earlier in the chat. We have some additional events coming up after that, and full details on all of those, including speaker lineup, is available at covidinfocommons.net. And finally, I've just dropped the link there to sign up for our mailing list in the chat as well. I know a number of you did that as part of your registration, and any of you who are signed up by next week should get a newsletter with full details about these upcoming events and the student paper challenge for your reference and to be able to share with your community. So I want to thank again all of our wonderful speakers today. Thank Helen and Florence for their contributions, and of course thank NSF for their support in this project. We are really glad to have built this community over the past year, and we're looking forward to more great things in 2021. So as Florence and Helen have mentioned, please do be in touch at info@covidinfocommons.net if you have any questions about anything we've discussed today. And until next time, thank you, everyone.
Bye-bye, and thank you, Katie. Thank you, everyone, and thank you to our researchers and Helen. Bye-bye.